This is my second talk for Meiolive, and this one is going to be a little shorter than the previous one. I would like just to give a brief update on what me and my colleagues are doing when it comes to phylogenomics of nematodes in general, and with specific focus on marine nematodes. So how far did we progress in this subject? And here's the team. We have five major people who are involved in this project. Kevin, Ashley, Nicholas, Mohammed, and me. All the names are correct. So let's see what it's all about. The history of that. One of the very first phylogenomic studies of all nematodes was focusing on parasitism, was published in 2014, and it only included 2 species of marine nematodes in this analysis. Now, going forward about 5 years, we published another phylogenomic study, and there were only 9 species of marine nematodes included in that. It's quite a considerable improvement, 4 times more than the previous study, but still very, very little. So now, this year, we are trying to gather all the published and newly generated transcriptomes and genomes in a single analysis. And part of that work is actually focusing on marine nematodes in particular. And here, what you can see on this graph is on the top, you can see the relative diversity of different groups of nematodes of different orders. So the width of each column is equivalent to a relative number of known species in the group. And then at the bottom, you can see the relative number of genomes or transcriptomes that are being included in this analysis. And you can see how biased the data set is that we have now. So some groups are very badly represented, some groups are represented by many data sets. And when it comes to nematodes that are marine, here's the trial was trying to highlight actually the number of genomes and transcriptomes of marine nematodes, and you can see how small the percentage of the data is available right now. I mean, not to say that general knowledge on diversity of marine nematodes is also kind of lagging behind the other groups like spirulina, animal parasites, plant parasites, and soil nematodes are studied much more actively. So we have multiple biases shown in this figure, not only bias in sequencing, but also bias in taxonomic focus. So now, next slide. This is the data set that I'll be talking right now. Only selected representatives, not all 300 genomes that we have available now in transcriptomes, but only 18 species. All marine nematodes that we have are marked in blue. There are not that many, 18 species of marine nematodes and some soil nematodes and few animal parasites and plant parasites used as an outgroup and just to represent other taxa in our analysis. So it's not fully biased. And the standard procedures here would be assessment of orthogroups using protein ortho, multiple sequence alignment of these orthogroups, cleaning the data from poorly aligned sites in each alignment. Then for each individual orthogroup, we infer the gene tree, run it through the phylogenetic pruner, which cleans up a lot. So it cleans up contamination, it cleans up paralogs, it cleans up multiple copies of the same ortholog. It cleans up, you can clean up sequences that are shorter than a certain number of bases. It can clean up sequences that are just too different from everything else. That will cause long branch attraction. It's a wonderful algorithm. And then what I did with these data sets, we ran phylogeny using RAxML based on the supermatrix. So all the individual gene alignments were merged together in one supermatrix, and we generated the tree. And as an alternative analysis, I would run individual alignments separately, create gene trees, and then merge these gene trees using two different types of software, Astral and Splitstree. And more about this a little later, but these are pretty standard methods. We are not, right now, we are not exploring different options. We are just using different software options. We are just using what actually works out of the box. So that's why our choices are like that. There are multiple choices of software that can be used for every single stage, but not all of them worked when we tried them. And that's how we ended up with this selection. So here you can see the phylogeny clades that are marked in blue, whatever, ultramarine, aquamarine are highlighting the marine taxa. The brownish color is terrestrial or animal parasites. And yeah, this is a tree generated with RAxML based on supermatrix. And everything seemed to be in more or less in agreement with each other, except for this Desmodora species, which is outside of where it should belong. It's classified in the order Desmodorida, but it seemed like it's long branched here. Then again, the Thoracostomopsis should be placed in Anoclida, but it's again outside of its clade. And then Ceramonema is placed also outside of the taxon where it's been classified currently based on morphology. However, I didn't mark it as red because this actual placement is in agreement with other non-morphological data and some morphological data, and we will look at it a little later. So this is how the phylogenetic hypothesis based on supermatrix looks like. Now, what Astral generated is slightly different, but still more or less the same. All these clades are well resolved and well supported. Ceramonema is in the same position. But Desmodora moved, which shows that there is something wrong with this species, either with the data or, and I can tell you right now, both Desmodora and Thoracostomopsis are actually transcriptomes that have very little coverage and a lot of missing data. So most likely these placements are caused by missing data. But what I wanted also to do in this case is just to see how much incongruence is between different gene trees and the species tree. And one way to do this is to generate these gene trees and merge them into a super network using Splitstree. And whenever there is an incongruence between different gene trees, it will create a network or connection in that part of the dendrogram. So this is not the actual network that was generated. This is a weighted kind of consensus network. It doesn't show all the incongruences. The original network was way too hard to interpret. But this is here, you can see that there are only two clades that are really well supported, and everything else is kind of problematic. All the other species, all the other taxa, they create this kind of network in the center, which just shows that there is no agreement between any of those individual gene trees. So by looking at the individual trees, I could kind of figure out what is going on. And what is happening in this particular case is that we have a lot of gene trees that are represented by a small part of species. We set up a limit. We didn't, we all the individual data sets that we included have to include at least 50% of the taxa. So, but many of these gene trees are represented by less than 100% of taxa. Another problem is that when we get these orthogroups, these alignments, through all the cleaning process with AliCut and AliScore and with phylogenetic, a lot of sites that have potential phylogenetic information gets removed, and we end up with alignments with very little information in them. The trees that we get do not have, are not well resolved, do not have, most of the branching is not well supported, and hence they are incongruent with each other. So one way of dealing with this that I'm trying to think if it's worth doing is actually removing those genes that are not resolving, are not producing the well-resolved trees, or tweaking the parameters of AliCut and AliScore to be less strict than what they are now. Anyway, here you can see the consensus phylogeny that basically between those trees, between the RAXML generated tree and the tree generated with AliCut, and we only have one place that's not well resolved between them. So there is a conflict in this branching between those two trees. So I created a polytomy. Everything else appears to be well resolved. How does it compare with 18S phylogeny? Well, most of the branching, some clades are resolved in the same way, like these groups here, these groups at the very base of the tree, while the rest of the tree in 18S phylogeny is not resolved at all, while we have a resolution here quite well. The reason for this is very simple. We are missing a lot of data from the genomic analysis that is actually creating a very complicated branching in this part in 18S phylogeny. For both orders, Areolemida and Monhistorida, in 18S phylogeny, they are represented by separate at least two well-supported clades, which are classified together, but they're definitely not related to each other. In our case, we only have representatives from one of each clade. So here is one of the points where we need to add more data. But again, here we have in phylogenomic analysis, we have the Muscalicida and Ceremonema grouping in the same clade. Here they are separate. Again, that can be a sampling bias, but what's more important is that both 18S and both phylogenomic analysis agrees in the fact that Ceremonema does not belong to where it's currently classified, which is order Plectida. As a conclusion to this little brief introduction, you can see that there is a lot of work to be done. A lot of data need to be collected, a lot of tweaking with the alignments. So this is what are we doing right now. We already have some extra sequences generated, some extra transcriptomes generated, which include 4 marine nematodes and 2 freshwater nematodes. By adding some of them, we will hope that it will help us place the Desmodora correctly in the phylogenetic analysis, and just improve the resolution of the tree. Of course, it's very, very little, so the idea is to get people, other colleagues involved who are interested in genomics of marine nematodes and generate more data for this analysis. We'll be looking at how to modify the data analysis pipeline so that we do not remove too much phylogenetically informative sites, but also do not introduce too many errors. And as I said, for some groups, we need to focus on generating transcriptomes for target species, not just random thing that comes in the sample or that's big enough, but for particular groups that are underrepresented in the phylogeny, are completely absent from our data set. And at the end, I would like to just acknowledge everyone and every organization, or nearly everyone and every organization that has been involved in this study, funding agencies, sampling locations or cruises, resources that we use to analyze the data, and all the colleagues and friends who have been involved in this project at every stage, from sampling, data generating, analysis, criticism, etc. And I would like to thank all of you who are listening to this presentation also. Thank you.